Here we'll discuss estrogen and progesterone. So let's start out with an overview of steroid hormone production. And remember that only certain cell types in the body express the specific enzymes that convert cholesterol into steroid hormones. And the rate-limiting step in steroid synthesis is conversion of cholesterol to pregnenolone, which is a key precursor of all steroid hormones. And so to make pregnenolone, the side chains are removed from cholesterol through the action of this cholesterol side chain cleavage enzyme, cholesterol desmolase, also known as P450-SCC. Pregnenolone is a precursor of all steroid hormones, but it's also important to note that progesterone is another important precursor molecule. And so within cells that have the appropriate enzymes, progesterone can be further processed to generate aldosterone, cortisol, androgens, or estrogen. So the ovaries are responsible for oogenesis and for the secretion of progesterone and estrogen. And the precursor for the biosynthesis of ovarian steroids is cholesterol, just like it is for all other steroid hormones elsewhere in the body. And so the general schematic shown in this cell over here is that enzymes in the mitochondria and smooth endoplasmic reticulum convert cholesterol into some steroid hormone depending on the enzymes that are present in that specific cell type. Then the steroid hormone diffuses out and into the blood. And so progesterone and estrogen are both synthesized in the ovary, progesterone in the theca cells and estrogen in the granulosa cells. So initially, the P450 enzyme, cholesterol desmolase, converts cholesterol to pregnenolone, and LH stimulates this reaction in the theca cells. Pregnenolone can then be converted to progesterone by an enzyme in the theca cells and then secreted. And pregnenolone can be converted through a series of steps and enzymes in the smooth endoplasmic reticulum of the theca cells all the way to androstenedione. And so androstenedione diffuses out of the theca cells to the nearby granulosa cells, which contain these unique enzymes. And it's through the action of these enzymes that androstenedione is converted to testosterone, and testosterone is then converted to estradiol. And so the activity of the aromatase enzyme that completes this final step in the synthesis of estradiol is stimulated by FSH. And so the activity of the aromatase enzyme that completes this final step in the synthesis of estradiol is stimulated by FSH. So if we zoom in on that interaction between the theca cell and the granulosa cell, each one of these cells synthesizes a different hormone because they contain different enzymes. So here is our big picture. We're looking at a developing follicle with the oocyte in the middle, the surrounding zona pellucida, and the granulosa cells in pink, and the theca cells out here in blue. And the theca and the granulosa cells are separated by this basement membrane. So now if we look closely at that interaction with these two cells here, in the ovary, the theca cells contain enzymes to produce androgens from cholesterol, but they don't have the aromatase enzyme that's required for that last step to convert androgens to estrogens. However, the granulosa cells do express aromatase, so they can convert androgens that are made in the theca cells into estrogens. And so this process of estrogen production happens in the early to mid-follicular stage. So estrogens are primarily synthesized in the ovaries, but also in other cell types. And so it's important to note that some cell types contribute to plasma concentration of steroids just by taking up one steroid and converting it to another. So adipose cells, for example, express high levels of aromatase, and they're able to sequester circulating androgens and convert them into estrogens. In addition, the skin, in particular the hair follicles and sebaceous glands, they're able to convert circulating DHEAs and androstenedione into testosterone, which may then be further processed into estrogen by the adipose cells. And then the liver is a major site of catabolism of steroid hormones. and it can also participate in the conversion of one steroid to another through metabolic processing. So estrogens are the main female sex hormone, and they're responsible for the control of functions in the female reproductive system, development of secondary sexual characteristics that appear during puberty and sexual maturity. And so if we focus on the natural occurring estrogens, which include estradiol, estrone, and estriol, almost all of the circulating estradiol is produced by the ovary through that two-cell mechanism we just looked at, with the theca cell producing androgens and the granulosa cells converting them to estrogens. In contrast, only about half of the estrone is produced in the ovary, and the other half is generated by conversion of steroidal precursors by cells in the periphery, like adipose cells or skin cells or the liver. And then finally, most of the estriol is produced by the placenta. This is the relationship between the gestational parent, the placenta, and the fetus. And the big picture is that there are steroidal precursors that originate from the fetal adrenal gland and liver. They're taken up by the placenta, converted into estriol, and then released into the circulation of the gestational parent. Now let's look at how estrogens bind their receptors and act in the cell. And so this is our round gray cell with the white cell membrane and the extracellular space out here. And so steroid hormones in general have a lipid-like molecular structure, so they're not very soluble in plasma. And so estrogen in the bloodstream is bound to proteins, like sex hormone binding globulin, for example. Like other steroid hormones, free estrogens enter the target cells by diffusing through the plasma membrane and binding to their receptors in the cytoplasm. That hormone receptor complex then enters into the nucleus and binds specific DNA sequences called estrogen response element, or ERE, which causes activation of transcriptional processes or signaling events that control gene expression. And so these cellular responses that are mediated by these changes in gene expression are known as the genomic effects of estrogen. Interestingly, estrogen, unlike other steroid hormones, has also been shown to have effects on its target cells that can't be explained by that nuclear receptor mechanism. And so this has been controversial, but now it's widely accepted that estrogen exerts some of its effects by binding to receptors that are on the plasma membrane and activating second messenger pathways. And so these are referred to as the non-genomic actions of estrogen. So now that we've looked at the synthesis of progesterone and estrogen, estrogen binding, we're ready to look at the physiological roles of estrogen and progesterone. And so one important role of these steroid hormones is preparing the uterus to support a fetus. And so histologically, the endometrium of the uterus is composed of glands with arteries and this stroma in the background that's made up of fibroblasts and extracellular matrix. So at the beginning of the menstrual cycle, the endometrium is relatively thin. But then during the proliferative phase, estrogen stimulates the growth of the endometrium so that it's much thicker by the time of ovulation. Then after ovulation, the ovary secretes progesterone, which further inhibits endometrial proliferation. Progesterone also has an important role in stimulating the endometrial glands to secrete glycopeptides and proteins to nourish the blastocyst, and stimulates both the glands and the spiral arteries to become more coiled. And so this phase is called secretory because of the increased activity of the glands. So then if there's no implantation of an embryo, the endometrium enters this degenerative or menstrual phase. If conception takes place, implantation can occur during that mid-secretory phase, at which time it's of sufficient thickness and full of sustenance. And as we discussed, there's then secretion of human chorionic gonadotropin, or HCG, which rescues that corpus luteum and thus progesterone secretion, which is essential for maintenance of the endometrium. At this point, it's important to note that the transition from a thin endometrium, typical at the start of the menstrual cycle, to a thickened endometrium that's typical of the secretory phase depends on the appropriate sequence of estrogen and progesterone released. And so estrogen has to be secreted first to stimulate proliferation of the endometrial cells, followed by progesterone to stimulate maturation of glands and the spiral arteries. And so this sequence ensures that the resulting endometrium is structurally stable and it's prepared to perform its supportive function if conception should occur. In addition to these roles, estrogen and progesterone have other physiological functions in both the non-pregnant and pregnant person. So the effects of estrogen on the uterus, in addition to that key role in endometrial proliferation, include its effect on the cervix to stimulate production of a watery elastic mucus, and also the sensitization of the uterine smooth muscle to the effects of oxytocin by increasing the expression of oxytocin receptors. There's also the effects in the ovary on the granulosa cells, as well as in the breast, where estrogen stimulates the growth and differentiation of the ductal epithelium. And the growth-promoting effects of estrogen in the breast and the uterus have been implicated in breast and endometrial cancer. In the liver, estrogen is catabolized, and so it also acts on hepatocytes to increase lipoprotein receptor expression, resulting in a decrease in serum concentrations of total cholesterol, increases in serum HDL cholesterol, and triglyceride concentrations. Estrogen regulates the hepatic expression of genes involved in coagulation, which plays an important role in limiting blood loss following labor and delivery. In the central nervous system, estrogen has neuroprotective effects, and in the bone, estrogen promotes bone maturation during childhood and adolescence and promotes the conservation of bone mass, in part by decreasing the formation of bone-resorbing osteoclasts. And as we discussed, the major physiologic roles of progesterone are in the uterus, where it stimulates glandular maturation and secretion during the secretory phase, and it also acts in the cervix to stimulate the production of a thick viscous mucus. Early in pregnancy, progesterone also facilitates implantation of the embryo and maintains pregnancy through stimulation of uterine growth and differentiation. In addition, progesterone acts on smooth muscle cells to inhibit contractility and maintain smooth muscle relaxation in the uterus throughout the pregnancy. And then in contrast, at the end of pregnancy, a decrease in progesterone levels enhance uterine contractility. In the brain, progesterone modulates sexual behavior and regulates body temperature. And increased progesterone levels during the luteal phase of the cycle increases both core and skin temperatures. Progesterone also affects lactation, so it stimulates the development of the mammary gland in preparation for milk secretion. It counteracts prolactin's effect in mid to late pregnancy, preventing milk protein synthesis before giving birth. And then the sudden fall in circulating progesterone that happens with birth is associated with increased prolactin secretion, so the removal of inhibition of milk formation, and then the onset of lactation.